So this is an outline of what I'm going to try to accomplish today. I'll start off talking about the importance of ovarian cancer, or the importance of metastasis. So I'll give you a little bit of an overview of ovarian cancer because it's probably something you don't think about on a daily basis. I'll go into some preliminary results of DNA sequencing studies, and I'll move into discussing the role of epithelial reticular transition in metastasis and in ovarian metastasis in particular. And I'll end up talking about some work we've been doing developing microRNAs as potential therapeutic targets. So ovarian cancer is sometimes called the silent killer. It's the most lethal of all the gynecological cancers. It's the fifth leading cause of cancer-related death among women. The overall 5-year survival rate for women with this disease is only 50%. And that statistic hasn't changed in 50 years. Ovarian cancer is classified according to the histology of the tumor. Ovarian epithelial cancers are the most common, 90% of all ovarian cancers are epithelial in origin. This includes serous papillary, which is the most frequent, 80%, and endometrioid mesothelioma. If the cells don't look like any of these types, then the tumor is classified as undifferentiated. Tumor stage describes how far the tumor has spread where it started in the ovary. So in stage 1, the tumors and cancers are confined to the ovary. Stage 2, they extend slightly outside the ovary to the fallopian tubes of the uterus. Stage 3 is when you start getting metastasis. You get microscopic implants throughout the abdominal cavity, specifically the most abundantly to the omentum and areas around the small bowel. And in stage 4, the disease spreads to other organs. You metastasize through the blood of the lymph system, you get to the brain and the liver, so it's spread all over the place. It's difficult to diagnose this disease because at early stages, it's essentially asymptomatic. So you don't know you have the disease in stage 1 or 2. Even at stage 3 and later, the symptoms are quite nondescript. Like you get abdominal bloating, feeling of pressure, pelvic pain, frequent urination, and so forth. So these are symptoms that are not atypical of postmenopausal women. People get older, a lot of us have some of those symptoms. So this disease is often misdiagnosed. A woman will go complaining of these diseases to the clinician. Only if the woman is persistent and if the symptoms persist as well will she get really looked at more carefully. So the disease is often not diagnosed until late stages. That's unfortunate because if you look at the survival rate, the disease could be diagnosed at stage 1 or 2, where it's confined to the ovary or the various media around it. It can be essentially cured with surgery. But the problem is it's not diagnosed at those stages. And when you get up to stage 3 or 4, you metastasize, you see the survival rate over 5 years drops precipitously. So the conclusion from this is that the high lethality associated with this disease is essential because of metastasis. All right, so what we're going to talk about today then is ovarian cancer metastasis and various potential strategies to treat this disease. So number 1, if we could identify the disease at stage 1 or 2, it could be cured by surgery. The problem is that the disease is essentially asymptomatic at those stages. And at this point, no accurate diagnostic test exists to determine if a woman has a disease at that early stage. So in a collaborative project with Facundo Fernandez and chemistry, we're working on metabolic-based test, which is very promising. But at this point, there is still no early diagnostic test. So there's a lot of women that will go through stage 1 and 2 completely undiagnosed, and the disease will be apparent at stage 3 where it cannot be solved with simple surgery. Right? If you get up to stage 3, then the next possibility of treatment is debulking the tumor, that is surgical removal of the primary tumor, the things that you can get, as you see microscopically. And then for residual disease, you have to treat it with chemotherapy. The standard of care currently for ovarian cancer is really a one-size-fits-all plan. First-line therapy is platinum-based drugs. which induce DNA damage, which hopefully then induce apoptosis or self-annihilation of those cells. And taxane, which is a drug that interferes with microtubules. So both of these, one is intended to induce apoptosis, the other to prevent division of the cells. The type of drugs used to treat the metastatic disease is determined by how long it's been since the patient was last taking a platinum drug. If it's less than 6 months, the patient is termed platinum resistant. And that is if you get recurrence within 6 months. It's just assumed you're resistant to that first-line therapy. And then it becomes a trial and error. Just try a series of drugs. So there's really no rational plan to it. It's just, let's see what works and what doesn't. If it's been more than 6 months, then it's assumed that you can go back to the platinum-based therapy. So the take-home from this is it's not very rational. It's really trial and error approach. The hope for the future, not only for ovarian cancer, for all cancers, really is to move to a genome-based personalized therapeutics, where we can feed in genomic information, DNA sequence information, expression information, protein level expression, so forth, and then through bioinformatics analysis to try to identify optimal drugs that would be effective for this particular patient. So each patient is treated as an individual as opposed to one size fits all. That's the dream for the future, and many people believe it's the way for the future. The one successful use of this approach that you may be familiar with is the which is an inhibitor of tyrosine kinase domain for the protein. So if a patient is diagnosed as having a mutation in one of these tyrosine kinase encoded genes, then they're prescribed Levac. Levac comes in and inhibits that particular region, and then it prevents the cells from dividing. And this has been an effective strategy for a number of blood cancers that we've seen. So this would be a paradigm that we would hope would come out in the future. So we recently started a project to develop a pipeline inputting RNA and DNA sequence data, and hopefully maybe also proteomic information coming in. And this pipeline is designed to go down and at the other end to come out with a predicted optimized therapeutic program for that particular patient. So we're trying to move into personalized medicine for ovarian cancer. And this is a collaborative project with Fred Amberg and Keith Jordan and myself, our labs, and talented people in those respective labs. You can see from his demeanor that Fred takes this project much more seriously. Probably because the team has tenure and Fred. All right, so the recent project we started is initiated with 8 patients for which we have samples of primary, metastatic, and recurrent tumors from each patient. And I want to acknowledge that these samples were obtained at Northside Hospital with the collaboration of the surgeon who works on ovarian cancer. The DNA sequencing of this is now going on in our lab. We're using an ion torrent system, and it's being done by DDET and Amelia in our lab. On average, we're getting about 200x coverage of DNA sequences. So what I'm going to just quickly go over today is the preliminary results. We have some data back on 2 of the 7 patients so far. So we can't make very many conclusions, but still the data is intriguing and interesting, so I wanted to share some. Okay. So this just shows the workflow. The computational work on this was, a lot of it was done by Siddharth, who's a master's program here in bioinformatics. We would like to have a say, but he may be leading, he's got office talker. He finds more attractive. And also, I'm going to talk about some of the preliminary analysis that was done by Roman Zenchev, who's a research scientist in my lab. All right, so the first thing is how genetically similar are the primary tumors for these patients. So if we look at the 2 of them, there's a lot more mutations. And I should say, I didn't mention it here, but when we do this, we collect samples from whole blood as well as DNA from the tumor. We use the whole blood as a representation of what that patient's genomic sequence looks like. So that any mutations that are variants that are there, we subtract them away from the tumor. What we're looking for in the tumor is a novo mutations that distinguish that mutation. Patient 2, I don't know if I think maybe didn't know this. They're above the age differences. They're 71 versus 59. The interesting thing here is patient 1 was put on no chemotherapy prior to surgery, whereas patient 2 was treated with carboplatin based drug for a period of about a year or so, or 6 months, 4 months, maybe 3 or 4 months prior to surgery. They often do this to try to debulk the tumor with chemotherapy, so it makes the surgery more effective. So it's interesting that we have 2 patients, one that was treated with chemo, one that was not. So when we look at the results, it's interesting that if we look at the number of variants or mutations in patient 1, there's a lot more variability in patient 1 than in patient 2. Presumably due to the fact that patient 2 was put on chemotherapy, and a lot of those cancer cells were killed or the samples. Interestingly, we found no common mutations between the two, but there was commonality in genes that had mutations. In other words, P53 was the gene that was mutated in both patients, not at the same position. Like both were not functional mutations. How genetically similar to metastatic and primary tumors? So if we look at the overlap, we see there between the primary and metastatic in both patients, there's some overlap. There's quite a lot of variability that's in the primary that does not show up in the metastatic in both patients. This large number, if we were dealing with bulk tumor, where you just take a hunk out of the tumor and some of the cells in there are cancer cells, and we call those contaminated cells. Sometimes when you do that, you can explain variability like this outside. But in our case, we're using laser capture microdissection or other specified dissection techniques with an effort to pull out only the infected cells of the tumor. So we don't think that there's a lot of contamination. There still may be some contamination, but we don't really think that this is due to that. What it means simply, probably, is that a subset of the cancer cells in the primary tumor are being transferred to the metastatic tumor. There's a lot of genetic variability that's in the primary tumor that does not show up in the metastatic tumor. What about these mutations? Could be due to de novo mutations, or more likely, perhaps, it's due to variants that are in low frequency in the primary tumor that were below our resolution to detect. Right? So both of those options are possible, and we'll, by doing a deeper sequencing coverage in the primary, we should be able to separate those possibilities in the future. If we plot that data out, so we're looking here now at the variant allele frequency of the mutations in the patient in the primary versus the metastatic. You see, again, evidence that there's a lot of genetic variability in the primary tumor present at moderately high frequencies. By the way, you see the frequency never gets higher than 50%. Anybody using teachings or anybody know why that would be? Why would the frequency of mutations never get higher than 50%? It's because we're talking about de novo mutations. I know you know the answer. That show up, and so you're a heterozygote mutation. And we're not talking about a segregated population. We're just talking about mutations. The only way you can get homozygous for mutations, you have a second mutation at the same exact position, right? And that's very likely to occur. So 50% is the upper limit. You see, there's quite a few mutations that are in fairly high frequency in the primary tumor. That means that cells carrying those mutations are in high abundance in that primary tumor. But there's a number of interesting things here. This is just showing the slope. I mean, there's a low Pearson correlation coefficient between the two, meaning that metastatic tumors are not simply representing accurately the composition of cells that are in the primary. And there's definitely an enrichment of variability in the primary. The interesting thing here is that we see a high frequency in the metastatic mutations that were in very low frequency in the primary. That suggests to us that clones or cells that are in the metastatic very low frequencies were the founders of the metastatic tumor, and therefore they go up to high frequency. So that's an interesting result. We find in both of these patients. I don't know if it's going to be generally true across all of the patients, but if it's random sampling, you would expect some of the high frequency mutation cells to carry over. So that's an interesting phenomenon that we'll have to follow up. You can take this kind of data and replot it, or you can just sort of clump together the number of mutations, which would be the number of cells at a given area in frequency, and plot it out this way, which kind of gives you the shape. So this says there's quite a lot of variability in the primary with high frequency mutations. There's a lot of variability because remember, over here are all of that variability that's present in the metastatic that didn't show up in the primary at all. That's when it's over here at 0. We think in the future, we might, when we do deeper sequencing, we can find a lot of these variants actually are in the primary, but at very, very low frequency. All right, so we can take those shapes and you can kind of infer how many clones may be present in the primary. So here it looks like there's at least 2 different types of clones of cells in the primary, some representing those that are high frequency. In the metastatic, we're seeing mostly low frequency, things that were in low frequency in the primary showing up here. This is again indicative that the metastatic site is being established from low frequency clones in the primary. A new algorithm has just come out. It hasn't even been published yet. It's in press that can take this kind of data and more accurately predict what's the number of different clones you've got in your sample. So in the primary, using this algorithm, it indicates that there's 5, at least 5 clones in the primary, and we see a subset of those clones being transferred to the metastatic. And it looks as though what's being transferred are low frequency clones. Okay, so the conclusions from this, both primary metastatic tumors are polyclonal, genetically heterogeneous. Genetic character of the metastatic tumor cannot be reliably inferred from the primary tumor, and metastases can be established from low frequency primary tumors. That has clinical significance, especially when you're talking about personalized medicine, because if you want to get the primary tumor and you want to determine what drug should be treated to prevent metastasis, it tells you you're going to have to know what are those low frequency clones. So you're going to have to see one early deep. You can't be satisfied with knowing what the most abundant clone is probably because it may be that that's not the clone metastatic. So this is all preliminary, but these are ideas that we got from this data that we'll be looking at further. Okay, another challenge for this whole problem with metastasis is the fact that metastasizing cells themselves may be different in kind from the primary tumor. So the classical view of metastasis is that the primary tumor sloughs off some cells, and those cells, say, integrate into blood vessels, another place, and then get transferred to another place and then start growing. Now, the presumption is there may be some genetic changes going on here slightly. For example, you're going to have to become less adhesive to the primary tumor to slough off. But in the classical view, there's no major change between the cells that comprise the primary tumor and the cells that are missing. In 2002, Jean-Paul Thierry proposed that metastasis involves major changes. It's not just a question of sloughing off the primary tumor. But he proposed that you are going through a process called epithelial transition, where the primary tumors are largely epithelial-like, and the cells somewhere in there, perhaps on the periphery of the tumor, undergo the epithelial-mesenchymal transition and become mesenchymal-like cells. They have a tremendous change in shape. They're more motile. They're able to invade more efficiently. And that is in that state you metastasize, and then you find a second site you attach, and you undergo a reciprocal event, mesenchymal to epithelial transition. These cells that are in the mesenchymal state are believed not to be replicated rapidly. So where they're more or less quiescent in terms of replication. That makes them less responsive to drugs that are designed to kill rapidly dividing cells. And so one of the things that he noticed, the notable things that people were noticing, is not only are these cells more motile and perfectly designed for metastasis, but they tend to be more drug resistant. So early on, several years ago, Roman and our lab tested. We have two cell lines, or a number of cell lines, some that are mesenchymal and some that are epithelial-like, very epithelial-like cells. We tested the sensitivity of these two cell types to cisplatin-based drugs. This is the IC50, which is the level of drug that will kill 50% of the cells. And you can see that the OPAR3 cells are much more resistant. They're dying at a much lower concentration of drug than the mesenchymal cells. So it appeared for ovarian cancer cells, it seems to be the same as what other people have reported for mesenchymal and epithelial cells. Thus, metastatic ovarian cancer cells may be escaping therapy and actually facilitate metastasis. Now, not everybody can, or even now, universally accept this idea that metastasis is associated with EMT. And most of the people, and perhaps the most distinguished person objecting to this is David Tarrant, who's a well-respected pathologist. And he wrote a paper called The Fallacy of Epithelial Mesenchymal Transitional Nucleation. It says there's no convincing evidence for the conversion of epithelial cells to mesenchymal cells. The biological repertoire in vivo. So there's a number of experiments in vitro where people have shown that these change. But in vivo, data is evidence is quite sparse. So he believes that the biological repertoire of normal and malignant cells is sufficient to account for the events observed in metastasis. So we wanted to know, could we find any evidence that EMT and MET is actually going on in vivo in ovarian cancer patients? So the first thing we did, we took those 7 patients and we took slides of the metastatic primary tumors to our collaborating board-certified pathologists over the woods, and we asked them, so can you tell us which ones are metastatic and which are not? He could not distinguish between them. As far as he could tell, there was absolutely no morphological difference between the metastatic and the metastatic. So that would be consistent to what Tarrant is reporting in pathology supremacy, and is what led them to the conclusion that there really is no going on in this stuff. But we wanted to look at the molecular level, and so Kia, former student in my lab, now she's gone on much higher levels of work at Emory as a postdoc here. She wanted to know, is there molecular evidence of EMT and ovarian cancer looking at these patients? So for these studies, we again use laser capture microdissection to capture the malignant cells from the two sample types. And we ran gene expression analysis using the after measures. And all of our gene expression analysis is done by me. And so, I don't know if all of you are familiar with heat maps, but this is just a way to sort of present the results of these data. We're looking, there was 3,365 genes that were significantly differentially expressed. The genes are down here, and each one of them goes across, and it's color-coded. It takes the average level of expression across all your samples, and it picks back a dark black color, essentially. And if the particular sample, particular gene, in that particular patient sample is overexpressed, it gives it a red color, and if it's significantly expressed, it's green. And so then it's clustering algorithms, take that data based on molecular profiles. So what we're looking at here is metastatic samples and primary tumor samples from a number of different patients. And so if you look at the results from this in the groupings, most of the patients we follow from the two, the metastatic and the primary samples clustered together. So the molecular profiles of the metastatic become look very good, which is consistent with the morphologic results. There was a group of a couple patients where metastatic samples showed that the expression pattern very different from the primary. We call those group 2. So So at first glance, you would say, well, the results of group 1 seem consistent with the hypothesis that no healing is going on, and the results of group 2 would suggest there are perhaps some changes, major changes going on. So we took that data then and put it into a functional analysis algorithm. So these are algorithms that look at pathways and ask, are there any genes differentially expressed that are overrepresented in particular pathways?
So you mentioned that the cells go through EMT and they probably also go through antibodies. And so why do you expect that once when you sample them?
You're right. And if it had completely gone back to epithelial, like you would not see the difference. And we're just hoping that we're taking a snapshot and get some evidence.
So if you see a difference, you'll infer that it's early.
So this data is not inconsistent with the EMT model, but it is consistent with the node. But look at, that's exactly what I did. So when we looked at the functional analysis pathway and asked which pathways are more representative of this, these are the top 20. And if we looked at it, you'll see 11 out of the top 20. In fact, 5 out of the top 5 are all pathways that are implicated in EMT. They're functional pathways that are associated with epithelial transition. So this suggests to us that EMT may actually be going on. And just as it's just noted, we looked not back at group 1 and group 2 for 39 genes that have been earmarked as known to be associated with EMT processes. And we're looking at differential expression. And so we see that in both groups, this is group 1, this is the group that showed no overall difference between healthy and metastatic. But if we focus in on EMT genes, there are indeed significant differences between the primary and metastatic in group 1 and group 2. And group 1 is a little bit less, which would be consistent with the idea that these cells are moving back to an epithelial type. But we're just looking at it. So rather than group 1 being different in kind from group 2, it's probably just a degree of difference of where they are in that trajectory going from mesenchymal back to epithelial. So we then did a heat map looking only at these EMT genes. And now we see that almost all samples metastatic were quite distantly grouped from their primary with these or these EMT-related genes, except for one patient. There was only one patient with these EMT genes that grouped together. So that, again, is exactly consistent with what Todd was alluding to. We're looking at a process that's in, you go through epithelial mesenchymal transition, you go to the new site, and then you start to go back to the phenotype and to the molecular profile of epithelial cells. So I think it's a transient process. So overall, we took this as evidence indicating that EMT is associated with ovarian cancer metastasis. All right, so if that's going on, and if these cells are less sensitive to standard drug therapies, how might we approach this from a clinical perspective to come up with better metastatic cells? So there are 2 lines of investigation that we're taking. One possibility is to identify new drugs that are effective against these mesenchymal cells. And the second is to identify ways to convert these mesenchymal cells back to epithelial-like cells. That is, how can we induce MET in these cells and then treat them with the drugs that are effective on epithelial cells. And so in our lab, we've tried both approaches. And Roman published a paper a couple of years ago where he took mesenchymal-like cells and screened them against about 800 drugs. Some of them established drugs, some of them experimental drugs. And he came up with a number of potential candidate drugs that are effective at killing these mesenchymal-like cells. I'm not going to go into any more detail on that. Some of these drugs, in fact, are already FDA approved for other applications of their off-label applications, but it seems potentially very effective at this. So, but he's published that in 2012, so I'm not going to go into that. What I'm going to spend most of the time talking about is work we've been doing where we find that microRNAs are useful in inducing mesenchymal epithelial transition. We can take these metastasizing like cells and by treatment with certain microRNAs, kick them back into an reestablish their sensitivity. All right, so microRNAs, if you haven't heard about them, are a group of small regulatory RNAs, or about 22 to 25 base pairs in limit, and they negatively regulate the expression of target mRNAs by binding to them, inhibiting the translation of the story. Human genomes believed to be encode over 1,000 microRNAs, and they're predicted to target over 60% of all mRNAs. So it's a major form of gene regulation. So this the connection between this started with examination of an epithelial cell line. So here you can see the phenotypic differences between the white cells and epithelial. Epithelial tend to be small, cuboidal kind of shaped cells. They rapidly divide, and the mesenchymal cells are more elongated, and they're, you can't tell here, but they're highly motile and so forth. So there's quite a different phenotypic between the two. If we look at the molecular profiling of these two cell types, we see these are biomarkers of EMT. So these are a subset of those 39 genes I talked about in the expression. And we see for, we're looking relative to part 3, we're looking at expression of T cells, white cells. The biomarkers that have been known to be represented in white cells are overexpressed, and biomarkers of epithelial cells are underexpressed. So everything looks good here. The interesting thing is when we look at microRNA profile, we found that several microRNAs were extremely intensively downregulated epithelial white cells, as opposed to and these mesenchymal white cells as opposed to epithelial white cells. So we thought, sort of naively, since it's extremely downregulated, supposedly ectopically express levels of these microRNAs, will they kick the cells back into the mesenchymal white phenotype? So it seems we weren't in any way thinking it really would happen. But we were lucky, and it did. So when we overexpressed one of those near 429, there's a negative control where we transfect the microRNA negative control sequence. We got no change. The cells remained phenotypically mesenchymal, but we treated with this 429, which was one of these ones that was extremely downregulated in mesenchymal cells. You can see we induced phenotypic change back to epithelial phenotype. And other microRNAs did not carry that well. So it was particularly 429 and other members in that family of microRNAs that were able to do this. We went on to look at this process dynamically over time points. So at time 0, we overexpress the transfectin near 429, overexpress it. And we're looking at the phenotype over a period of 144 hours. And this work was done by it's hard to see here, but here's the mesenchymal-like phenotype, that long, elongated type. At 24 hours, it still looks pretty much the same. By 48 hours, though, we see some dramatic changes in the phenotype. And then, if left without any additional transfection, when we get down to 44 hours, the phenotype is returning back to the mesenchymal type. So it's a transient kind of change that we're getting by reducing this microRNA. This is just close up at 48 hours, so you can clearly see the difference in phenotype between the negative control, which remains elongated, and then the self-treated here. This is what it looks like 144 hours. You can see the phenotype is coming back. It looks very much like it was. We then went on to do gene expression profiling of this, and Lucia again did this initial analysis. You can see right away that if you take 0 hours, the most dramatic molecular changes are apparent at 48 hours, which is exactly the time point where we see the phenotype changing dramatically. And then the molecular profiles begin to go back to looking 144 hours was more somewhere in between 24 looking at 48, see the red starts to show. So both phenotypically and on a molecular level, we demonstrate this is a transient process that this microRNA is able to keep the cells go from zinc type. More recently, she just published last month, Netta, who's a graduate student in my lab, showed that this effect is not only limited to mere 429, but can be great by any member of this family of microRNAs can bring about this phenotypic change. And with the similar changes in expression, and what's probably interesting from a potential clinical perspective, a significant drop in sensitivity, a significant gain in sensitivity to drugs, coincidental with the change in the phenotype in molecular genes. It would also be trans. It would also be trans. So when you go up to 140 hours, so that's all interesting. But these were cell lines established in the lab. So we wanted to see, does this hold up in actually metastasizing cells from patients? So we established recently a primary cell line from a patient, and this was established from metastatic cells isolated from the cytosol fluid, late stage ovarian cancer patient. And so, she's done a number of characterizations of the cell lines. I'm not going to go into this. But these cells, when they're grown in culture, form steroids, which is typical of mesenchymal stem-like cells. So it has features of stem-like cells in that regard. When they're grown on plates, they grow and show the phenotype of mesenchymal cells. Well, this is just that cell line, which is the mesenchymal cell line, and the old cartridge cell lines that we were studying in the lab. So you can see that the phenotype of these cells is very much like when she did ectopic overexpression of negative control in these cells, there was no change in phenotype, but again, if we overexpress 429, we see a change into a more epithelial phenotype. The biomarkers, again, show indications that after transfection with 429, we're seeing changes in decreasing biomarkers representative of the epithelial cells. So everything looks like it's in the mesenchymal epithelial transition in the cells, just like it did in the cell line. And then, perhaps most interestingly, the sensitivity to cisplatin again significantly increased in these cells after we induce them to go through this transition. So this suggests that this may be a potential clinical benefit to patients being treated with these microRNAs. Could kick these metastasizing mesenchymal cells back to a phenotype, even if it's transient. It gives you a window of opportunity to then treat those patients with standard cisplatin polyplatin-based therapies, and that may help prevent metastasis.
John, are those epithelial biomarkers direct targets of the microRNA?
Some of them are, and some of them are not. So there's a lot of indirect regulation going on. So it's estimated for these microRNAs, direct targets are in the order of around 200 genes directly targeted. When we look at gene expression, we see thousands of genes. So there's a lot of indirect effects.
What about the effects of those microRNAs on non-tumor cells off the target effects?
So, well, if we've tried, for example, taking epithelial cells, and if we could put integramers of these microRNAs in, and we get nothing. So it doesn't work both ways. There's no noticeable effect of this on if you transfect these old cartridges. So, but it does work on other mesenchymal cells. We've tried glioblastoma cells, and so we seem to be able to get changes as well.
I just wanted to follow up on King's question. Since the microRNAs work post-transcriptionally, have you evaluated the post-transcriptional effect of targets, or have you just looked at the gene expression levels?
So, well, at this point, we're just looking at RNA levels, so we're not distinguishing between those two. We could go in and do that at some point by looking at stability.
You probably know that that family is so huge in the development of epithelial type organs.
Which family? These microRNA? It's a 200
the
family,
200
so
family,
there's only 5 members.
They target things like SOX2, Notch,
Yeah.
PIDX2, things that are necessary for the development of that field.
Now, the primary target that people have been talking about with these is SAB, because because it's a direct target of this, and Z is a repressor of a number of epithelial markers. We have done experiments where we've gone in and treated with knockout of Z with siRNA. We do not get this phenotype. So it gets very complicated and moves you into the area of systems biology because it's not a simple one-to-one kind of thing. There's probably a lot of genes involved with alternative pathways. This is one of the problems you have when you're dealing with these cells is because over evolutionary time, you're going to evolve backup systems, and the cells are very robust. So trying to dissect the actual action of these things is going to take a while. So to summarize what I've done today, then, ovarian cancer is an extremely lethal disease. It's typically not diagnosed until after metastasis has occurred. The primary metastatic tumors are polyclonal. They're highly genetically variable mutations. It indicates the importance of using personalized medicine. Metastatic tumors can be genetically divergent from primary tumors and may be established by clones that are low frequency in primary tumors. Ovarian cancer metastasis is critically associated with the gene and is molecular parasite cells are resistant to primary cancer. Ectopic overexpression of this family of micronases emerging as a second strategy to induce ADT hairline disease and metastatic cells. So I'll stop there and think additional questions.
You said there was no complications in the primary
So what we're looking at there are only ones that were in common.
So there were some complications.
So the first thing I was comparing patient 1 to patient 2. There's no complication between primary and metastatic.
Okay, so then I have a question. You said you see these in the metastasis that has metastatic cancer, which is low frequency. Primary, that means you went into the metastasis. So the patient is high.
Right. So that's a great point. That if you think about it, those mutations that are in high frequency are presumably ancestral of in the primary.
But low frequency is
In the primary, low frequencies would be new mutations. If a low frequency, the cell with low frequency mutation goes to the metastatic, then it starts populating.
exactly.
Then it'll go to high frequency.
So this person occurs late in the stage.
Oh yeah. Yeah. Go ahead.
Okay, so based upon this, so the lesson in the cells that are like stem cell like tissues, is there anything more about what triggers this?
What triggers it? There's some people that think that it's going on all the time. So there's lots of theories. One could say there's cancer stem cells embedded in the primary tumors, and it's a subpopulation of low frequency. It's possible that metastatic cells are coming from both. See, that's an intriguing hypothesis that would be consistent with the fact that low frequency mutations are the things that low frequency clones are the things established. No, that's right. So it would be variable because they're in each patient is going to be generating variability going on. So it's that. The other option is that on the periphery of the tumor, somehow there's signaling coming in that stimulates those epithelial cells to go to immunity, and then those cells. So it's not known exactly.
In the sloughing model, the bloodstream is the main pathway of the cells from the primary to the metastasis because the cells are motile. But once the cells undergo EMT, then they become motile. They become almost like little breast cells, and they can migrate through tissue. But it looks like in that model, they're still sort of going through the bloodstream. And I wonder if does that new model account for the fact that these cells can basically be moving through tissue?
Moving what?
Tissue. Through tissue.
Through tissue.
So
yeah, moving through tissue and also being able to get into the blood vessel. We're doing some collaborative work with Michelle Dawson and other people that are looking at the physical properties of these cells, and they're much more flexible, and they're able to sneak into smaller things. So people think that the zinc lung cells are much more prone to get into the blood vessels and lymph system than our epithelial cells. So that's one thing. But the other thing, of course, is the fact that they're resistant to chemotherapy. In our case, most of the metastasis is not being transferred through the lymph system. It's just cells coming off the primary tumor and spreading within the abdominal cavity to other sites. But there's clearly also movement going through the lymph or blood system because you get tumors in the brain as well in some late-stage patients.
So given the number of targets that microRNAs have, are they viable as therapeutics, or would you have to identify a subset of targets and develop siRNAs?
So the interesting thing about microRNAs, if we look at all of them, Chris Hill in my lab has been doing this, is that all these microRNAs will target about 200 genes, but if you look at what genes they're targeting, there seems to be consistency among the type of genes. So mere 200 families seem to be favoring genes involved in EMT-type processes. Other microRNAs seem to specialize on controlling cell cycle type things. So over evolutionary time, these microRNAs have probably evolved. So even though it's 200 genes, they seem to be representing in non-random fashion genes involved in a particular type of process. So that's...
You mentioned that the mutations which are found in the metastatic tumors are rare in the primary tumor.
Some of them.
Could that be just largely due to the fact that it sounds like the things that are metastasizing are somewhat weak and they don't grow as fast. So there might be a selective disadvantage within the tumor, but they have a sort of between tumor, disadvantage.
It's possible, but they convert back to epithelial cells in the end here. Lots of things are possible, unfortunately. We're just looking at this phenomenon and we're coming up with hypotheses. But I think it's wide open.
With the mutation analysis at the beginning, when you're saying there's no real common mutation between different patients, does that mean there's not common at that gene? Or at that point?
Yes.
But could there still be specific P50 or certain proteins that are certain?
There are. There's 5 overlaps. The other thing, remember these 2 patients, one of them was pre-treated with chemotherapy. So that may have killed a lot of the cells. If we look at 2 patients that were not pre-treated, we may see so it remains to be seen. But what we do know is different mutations in different patients are very, very different. So I think this one-size-fits-all is not really an effective way to go. Personalized medicine will be the best way to go.
So John, one of the things that strikes me about this is we've been working on cancer a really long time, and you keep saying it's wide open. It is, there's so many things we don't understand. What do you think we need to do to make advances that are significant?
So, I'm not sure that we'll ever cure it. I think we can get it to the point that maybe it becomes a chronic disease, like arthritis. So maybe what's going to happen, and this is pure speculation, but we will identify mutations in the tumor when we treat those genes, and recurrence may occur because we can't identify all of the subclones that are in there probably. And so the patient will have to be monitored. If it gets recurrence, you go and do the same thing and you treat again. So it may be a disease that can never really be cured, but one that can be contained such that you'll die of something else before you die of the tumor. I would think that would be our best hope. Anything else?
Any other questions? Thank you, John.